Aquatic ecosystems around the world regularly receive pollution from byproducts of energy production. Whether that's runoff from coal ash or accidental release from nuclear power plants, our aquatic ecosystems and the animals that call them home can face dire consequences from this contamination. This can include everything from individual mortality to population level impacts, which can result in the loss of keystone species and reduced biodiversity. Over decades, water quality guidelines have been established to protect our waterways, not only for our own use, but also for these vulnerable aquatic species. But are they truly suitable for one of life's most unique processes? Metamorphosis is a process many aquatic insects and amphibians, like frogs, go through. This can involve complete rearrangement of internal organs and growing new limbs. Animals that go through metamorphosis also often have a change in environment, like frogs adapting from aquatic environments as a tadpole to terrestrial as an adult. Insects and amphibians play a huge part in the food chain, and negative effects suffered from pollution exposure during their aquatic stages could result in repercussions all along the chain. There is a gap in knowledge around the effects of many metal pollutants on metamorphosis. Is it sped up or slowed down? Are the animals able to eliminate these metals, or are they retained and potentially transferred on to their predators or their offspring? My research aims to address this by using radioisotope tracing techniques to be able to follow the uptake and loss of minute concentrations of metals in frogs and insects as they go through metamorphosis. These techniques will also allow us to image exposed animals to determine exactly where any remaining metals are concentrated in the body. By determining just how much metal is lost or retained and what specific effects that may have on these animals, we can work out if our current water quality guidelines are fit to protect them. An important part of this process is also ensuring that our lab studies are representative of real-life situations. So my future research will also involve a field case study at the Savannah River site in South Carolina. This is a site that's aquatic ecosystems have been contaminated by decades of nuclear weapons production and a coal-fired power plant. Carrying out field experiments there will ensure that the results of my lab work are applicable to natural systems that have been polluted, and will help determine just how effective potential remediation of these areas will be. It's essential that along with understanding the effects of these contaminants, we're also able to plan how to mitigate or remove them and have in place guidelines that we know are suitable to ensure the ongoing protection of aquatic organisms at risk.